Hello viewers, today we will discuss the drug metabolism, and our today's learning objectives are that by the end of the session you will be able to discuss the mechanism of metabolism, that is what are the mechanisms behind the metabolism, then the very important phase 1 and phase 2 reactions and its clinical importance, how they are clinically important. Then enzyme induction and inhibition, the topmost mechanism behind the drug-drug interaction, especially in case of the polypharmacy, and some of the drugs they induce and some of the drugs they inhibit the metabolic transformation. And then the clinical importance of this metabolism as a whole and how this metabolism is clinically important. So these are our learning objectives. Before going to talk about drug metabolism, now the question must arise that what is the need of metabolism? Why the drug metabolism is needed? The answer for this question is that there are certain biological activity of the drug is important. Certain drugs require the biological activity. These are the prodrugs. The prodrugs will be converted into the active drug is one of the important reason behind the metabolism, and some of the drugs they are converted into the active metabolites, so their activity is being enhanced after the drug metabolism. And then to terminate the action of drugs, it's very much important to terminate the action of drugs, and it requires the metabolic biotransformation. Without the metabolic biotransformation, the termination of drugs is not possible. Termination of action of drugs is not possible. And then to convert the fat soluble drugs into the water soluble drug, because fat soluble drug will be reabsorbed from the proximal convoluted tubules of the kidney back into the systemic circulation. So for the excretion of the drugs through the kidneys, the fat soluble drugs must be converted into water soluble drug. So for the proper excretion of the drug, the biotransformation of this fat soluble drug into water soluble drug is very important, and this is one of the important function of drug metabolism. And then if the drug is already water soluble, so these biotransformations make it more water soluble. So for these reasons, the drug metabolism is important and is required. Now, there are 4 possibilities that if we administer the drug orally and the drug is water soluble, that this yellow color of the drug, blue color of the drug shows the water solubility. That if the drug is water soluble, the route of administration is oral, it reaches to the gastrointestinal tract. From the gastrointestinal tract, it reaches to the portal circulation, and from the portal circulation, it reaches to the liver. And from the liver, it reaches to the systemic circulation. And this water soluble drug, which becomes more water soluble in the liver after the biotransformation, is now available to the kidney for the excretion, and the 100% of the drug will be excreted out. What is the another situation? In the another situation, if the drug is fat soluble, the route of administration is oral, it goes to gastrointestinal tract, and after absorption, it reaches to the portal circulation, and the drug converted into the water-soluble drug after metabolism in the liver, and the 100% of the drug is converted into the water-soluble drug, reaches the systemic circulation. The same water-soluble drug is now available for the kidney for excretion, and 100% of the drug will be excreted out. In third situation, if the drug is fat-soluble, route of administration is oral, it reaches the gastrointestinal tract, it absorbs into the systemic circulation and then into the hepatic circulation. It goes to the liver for the metabolism, and what happens that half of a drug is metabolized and converted into the water-soluble drug, and the half of a drug is remain unaltered as the fat-soluble drug. So the 50% of a drug is water-soluble and the 50% of a drug is fat-soluble in the systemic circulation. And when this drug reaches to the systemic circulation and reaches to the kidney for the excretion, that only the 50% of the drug, which is water soluble, is available for the excretion and excreted out through the kidneys, while the 50% of the drug, which is fat soluble, again reabsorbed back and reaches to the systemic circulation, and again there will be the recycling of this fat soluble drug. So this is the another importance of this drug metabolism, that if the drug is fat soluble, it could not be excreted out through the kidneys until and unless it will not convert it into the water soluble drug. And what is the other situation? In the other situation, the drug is fat soluble, route of administration is oral, it reaches to the gastrointestinal tract, it absorbs in the systemic circulation and then reaches the portal circulation or the hepatic circulation, and it goes to the liver, but none of the drug molecule is metabolized into the water soluble drug. Without metabolism, the same fat soluble drug reaches the systemic circulation and it is being reabsorbed back from the proximal convoluted tubule, and none of the drug is available for the excretion from the kidney. So again there will be the reabsorption of 100% of the drug, and there is, so the duration of action of this drug is more and more, and so it might produces the adverse effect and the side effect or the undesired effect. So this is the another clinical importance of drug metabolism. Now, drug metabolism follows two elimination kinetics. That is the first order elimination and the zero order elimination. And metabolic transformation of the drug is catalyzed by the enzymes. There are certain metabolic enzymes which are required for the drug biotransformation. So in the first order elimination, if the dose of the drug is less than the enzyme capacity and the enzyme is not fully saturated, then the drug obeys this first order elimination. That is, the constant fraction of the drug is being eliminated out per unit time. Constant fraction, that is the 50% of the drug at this particular time period, then another 50%, then another 50%, and then another 50%. So this is the constant fraction of the drug is eliminated out per unit time, and the majority of the drugs, they follow this first order elimination. If the dose of the drug or the concentration of the drug is more than the enzyme capacity, and the enzyme is fully saturated or sometimes oversaturated, then the drug will follow the zero order elimination. And what is the zero order elimination? That is, the constant amount of the drug is being eliminated out per unit time. And you can see, for example, if we administer the 600 milligrams of the drug and the metabolizing enzyme is fully saturated or oversaturated, so the constant amount of the drug is being metabolized or eliminated out per unit time. Usually, the drugs follow the first order elimination and there is no effect of the dose or the concentration on the plasma half-life. But for some drugs like aspirin, like norepinephrine, when in small doses, they follow the first order elimination. But if you increase the dose of those drugs and the enzyme will be fully saturated, then the first follow the zero order elimination. So their concentration is decrease, and decrease. And when the concentration reaches below the saturation point of the enzyme, then again they follow the first order elimination. So in this situation, for those drugs, if we increase the dose of a drug, their plasma half-life will be increased, that as in case of aspirin and the phenytoin. Then these are the two well-defined metabolic phases. If we administer the drug first follows the first phase, that is the phase 1 reaction. That in the phase 1 reaction, either it introduces or it unmasks some functional groups, and the metabolites do not excrete it out through the kidneys so that they go to the phase 2 reactions. So this phase 1 reaction is either the drug is activated or it might be unchanged. And this phase 1 reactions, they are the oxidation, reduction and hydrolysis, and then this follows to the phase 2 reactions, and then these phase 2 reactions, that they are the conjugation reaction, and they are converted into the conjugated metabolites. But there are certain type of drugs who doesn't follow this sequence, and they first go to the phase 2 reaction, and then after conjugation they goes to the phase 1 reaction. So this is for the very few drugs. Otherwise majority of the drugs, they follow first the phase 1 reaction and then they follow the phase 2 reaction, that is a conjugation reaction. But there are certain type of drugs who follow first the phase 2 reaction and then they follow the phase 1 reaction. Now these are the phase 1 reactions, that is the oxidation, reduction and the hydrolysis. And in case of the oxidation, we have the very important subdivisions, that is it is the P450 enzyme system dependent oxidation or the P450 enzyme independent oxidation. And they have a very important place, that this P450 is cytochrome P450 enzyme system, that is the mixed function oxidase system, has a very important place for the oxidation of different type of drugs. And then this is the hydrolysis, that for the hydrolysis, that certain type of esters, they metabolized by the hydrolysis reaction. So for example, the succinylcholine is an ester that is metabolized by the plasma cholinesterase enzyme, and this process is very rapid, and duration of action of a single dose of succinylcholine is about 4 to 5 minutes, and in some people even they are less than 4 minutes. Some people has abnormal form of this enzyme, and that they more slowly metabolizes the succinylcholine and other esters. So in such condition and in such individuals, that is the neuromuscular paralysis may be produced by a single dose of succinylcholine, because the pharmacological effects of the single dose of this succinylcholine persists for even more than 1 hour, or in some people even more than 1 hour. So this is the clinical importance of this phase 1 reaction, and this P450 enzyme system has a very important place for the metabolism oxidation, and there are the certain type of drugs who enhance this metabolic activity, and those drugs, they are known as the enzyme inducers. So enzyme induction is usually the result from either the increased synthesis of P450 enzyme, or it might be due to the decreased degradation of this P450 enzymes. So these drugs, like the very important drug inducers, that is the carbamazepine is there, and then the phenobarbital, that is long-acting barbiturates, they are the very potent enzyme inducers, and then there is the phenytoin, the anti-epileptic drug, that, and other is rifampin, that is the antibiotic used for the tuberculosis, treatment of the tuberculosis. So these are the very potent drugs who are very potent enzyme inducers. So these drugs, they accelerate the metabolism of the other drugs if they are being co-administered. So they decreases the pharmacological effects of co-administered drugs, they decreases the plasma concentration of the active drug, or they increases the biotransformation of the other drug when they are being used with other drug or being co-administered with the other drug. So in case if the drug metabolically transform into the reactive or the toxic metabolites, then it definitely causes the metabolite mediated toxicity, like acetaminophen. It causes the metabolite mediated toxicity. The drug activity may be increased, that in case of diazepam, when if you co-administer the diazepam along with the another enzyme inducer, so the diazepam will be converted into the active metabolites like nordazepam and oxazepam, which have even the more potent sedative and hypnotic effects, even more than the diazepam. And in a way that the diazepam effects, they are being enhanced, and the duration of action of this diazepam is being enhanced. Another group of the drugs that is under the heading of the enzyme inhibitors, that inhibits the enzyme, that is the P450 enzyme, and they are called the enzyme inhibitors. The cimetidine is a very potent enzyme inhibitor. It inhibits the enzyme P450. Isoniazid, again the antitubercular drug, and then another is the xylitol, that they inhibit the lipooxygenase enzyme and inhibit the synthesis and generation of the leukotriene, and then that is the xylitol-leucost, that is the leukotriene receptor blockers. And another important is the ketoconazole, very potent. That this cimetidine and this ketoconazole is a very potent type of the drug inhibitors, and actually these drugs, they bind tightly to the P450 enzymes like a competitive antagonist, and these drugs, they efficiently and effectively decreases the metabolic activity of this P450 enzyme system to the substrate or the co-administered drug. So like omeprazole, that is the proton pump inhibitor, that is a very potent enzyme inhibitor. If we administer this omeprazole along with the warfarin, that is the anticoagulant drug, and if these two drugs are taken together, or there is the co-administration of these two drugs like an omeprazole and the warfarin, so what happens, that plasma concentration of the warfarin will be increased, and the pharmacological effect of the warfarin will definitely increase, that the inhibition of the coagulation system is very much increased, and it might lead to the hemorrhage. So this is the clinical importance of this enzyme inhibition and the enzyme inducers drugs when they are being administered or co-administered with other drugs. Now these are phase 2 reactions, and in this phase 2 reaction, that is the glucuronidation, and very important is the conjugation reactions. Most important phase 2 reactions, they are the conjugation reactions, and that is the glucuronidation or glucuronyl conjugation. There is the glutathione, glycine, sulfate conjugation, and this is the acetylation. Is and the drugs like isoniazid, anti-tubercular drugs, and this is the procainamide, it is amide type of drugs. They metabolized by this acetylation, and this isoniazid and procainamide are inactivated by N-acetylation, and that is some of the individuals, they are deficient in acetylation capacity of the enzyme, so they are known as the sialoacetylators, and may have the prolonged and the toxic effects of the drugs to the normal doses, even or sometimes less than normal doses. So these sialoacetylators are about 50% of white and African Americans in the US, and much smaller fraction of the percentage of the Asian population. So sialoacetylation trait is inherited as an autosomal recessive gene, and this is one of the reasons of drug-induced adverse effect, because when the acetylation, the very potent important type of an phase 2 reaction, it slows down, so the drug reduces the adverse effects, and the duration of action will be more and more. Another very important phase 2 reaction is the conjugation reaction, that is the glucuronidation reaction. Apart from the metabolism, this has a very important place for the drug-drug interaction. Like this is the drug, like those drugs, highly lipid-soluble drugs, like corticosteroids or the steroidal type of drugs, like an oral contraceptives that we use orally, that is highly lipid-soluble drug, they or first goes to the gastrointestinal tract, and because of the lipid solubility, they absorb to the systemic circulation. And when they absorb to the systemic circulation, so from this systemic circulation again they reaches the hepatic circulation, and they get access to the liver. And at the liver, this highly fat soluble drugs, they are converted into the water soluble drug by the glucuronidation or glucuronyl conjugation. That there is the conjugation of the glucuronyl group, and this fat soluble drug converted into the water soluble drug. When this drug is converted into the water soluble drug, then they are eliminated through this bile and this common bile duct into the intestine, and these are the this is the drug, that is the conjugated drug that is available in the intestine. And this is the route of elimination, that the water soluble drug is get eliminated out through the feces. But in the GIT, gastrointestinal tract, we have the normal flora, and this normal flora, the normal growth of the microorganisms, they releases the enzyme, that is the glucuronidase enzyme. And what is the function of this glucuronidase enzyme? That it deglucuronidases the drug, that the drug again become the active, again become the fat soluble, and again the drug is available for the systemic circulation, and it reaches the systemic circulation, and again from the systemic circulation it gets access to the liver, and again there is complete recycling of the drug will take place. And that's why we call it as the enterohepatic recirculation or enterohepatic cycle. That is, it is the cycle between the anterior GIT and hepatic, that is the liver. Now, what is the clinical importance of this enterohepatic recirculation? That the duration of action will be prolonged. For example, if we talk about the oral contraceptive, they have a prolonged duration of action. So they have the very prolonged cover to the person as far as we are talking about the contraception. But if the lady who is having this oral contraceptive is suffering from some acute infection, and we prescribe the broad spectrum antibiotics for those ladies who are already having the oral contraceptive, then what happens? That these antibiotics, they disturb the normal flora, and they do not only disturb the normal flora, but they decreases the availability of the glucuronidase enzyme, which deglucuronide this drug, and the drug will become again active. So what happens, that the duration of action of a drug will be decreased, and the elimination or the excretion of the drug through the feces will be more and more. So the contraceptive cover to the lady will be shorter and shorter, and it is the clinical consequences that she might get conceived even after having the oral contraceptive. That is the very common clinical complaint by the different females. So if your patient is already having the oral contraceptive and you are going to prescribe any antibiotic, especially the broad spectrum antibiotic, you should educate at least your patient that she definitely get some extra measures as far as the contraception is concerned. So this is all about the clinical importance of this metabolism, that we have done the phase 1, phase 2 reaction, and this phase 1, phase 2 reaction, and especially the P450 enzyme system oxidation, mixed function oxidase enzyme system. They have a very important place for the drug-drug interaction is concerned. Enzyme induction and enzyme inducers drugs, the co-administration. So it's very important, and we have to adjust the dose when we co-administer these drugs, whether the drug is enzyme inducer or whether the drug is enzyme inhibitor, to produce the normal pharmacological effects of the drug. Thank you very much.